are starting now on what I like to call sort of the biochemistry portion of the course, okay? And while it is still chemistry, it is very much biology related and medical sort of related stuff, okay? And there's a lot less math on this portion that we're starting on now, okay? So if you are really good at memorizing stuff and you like that sort of thing better, congratulations. If not, sorry. And curious, I keep getting asked, like, what part is the hardest part of this course? There is not a hardest part for everybody. Like, I have equal numbers of people that kind of do bad on every exam and do well on every exam. It's just, this course just covers a lot of different sort of stuff. And so different people have different strengths, right? And will do better on certain exams. Okay. All right. So last time we've talked a little bit about amino acids. We're going to specifically talk about proteins. These are sort of your workers of your cell. They kind of do everything that you think of as you and actually doing things. Okay. So proteins are going to be what are called polymers. Poly means many, meros means parts. And so proteins will be composed of recurring things that we call amino acids, chained one after another, and then folded up into structures. Okay? And so they are polymers, repeated units of amino acids. Okay? You'll sometimes refer to them or hear them referred to as polypeptides. And that's just poly meaning many, and then peptide meaning the linkage that connects the amino acids. So there's going to be 20 different commonly occurring amino acids, and proteins can contain as few as 50. They can also contain as many as thousands of amino acids. So they can be, not small, but smaller, and they can be very large. Okay. In fact, you have proteins in the form of your hair and nails that you're actually seeing long polymers that are continuously chained. They're like complete polymers right here. Okay. Now, the ordering of the amino acids in a protein, this is going to determine both its structure and biological function. It's going to be a very important part of that. Though not the complete part, you do need it to be ordered in a certain way or within a certain category. But it can fold slightly different and make slightly different things. Now, the ordering of the amino acids, again, very important. The proteins, these can do all sorts of things. We've talked about hemoglobin. That was a protein that can transport oxygen in your blood. It's going to conserve the main components of skin and muscle that actually gives you the structural strength component of that. It's going to defend our bodies against infection. It's going to direct cellular chemistry. That would be enzymes that do those, where it actually makes the reactions faster. Remember, enzymes are biological catalysts. And they'll control metabolism as hormones as well. So most of the things as you think of as being human and doing stuff, most of them are proteins. There aren't all of them, but a large quantity of them. Now, the amino, this is actually going to be for the protonated amine portion. And so that's the NH3+, that's the amino, as you may recognize from our organic naming section, right? And then the acid part is going to be for the carboxylate, which is the conjugate base of carboxylic acid. And so the acid part's for the carboxylic acid, but because we're in physiological pH, we're in the bloodstream usually, we're going to lose the H+. So it'll be the carboxylate. Okay. Now, in the amino acids, these are groups are going to be bonded to a central carbon. This is going to be referred to as the alpha carbon. And that's because it's alpha position, which is the next one over, from these two functional groups. Okay. And so it's attached, it's next one over to the two functional groups right there. And so right here, we have the amino, the protonated amine right there. Here we have the carboxylate ion, that's the carboxylic acid, the acid part, so amino acid. Right here we have the alpha carbon. See how it's the one over from both of these? It connects them both. Okay. Over here, we're going to have the final component of an amino acid, and that's going to be the R group. The R group is where all the amino acids will change, and it's why we have 20 different ones. Okay? Is because we have 20 different R groups that are going to be commonly found. Does that mean that's all the amino acids that are possible? No. No, you have lots more that would be possible. They just don't commonly get used in animals because life has developed to use the 20. Okay? Primarily. Now, the one last thing I haven't talked about is this hydrogen right here. There's a hydrogen on the alpha carbon. It needs 4 bonds. I know. We just don't care about hydrogens, do we? Yeah. The last chapter was the last chapter we actually cared about hydrogens. Hydrogens had their time in the sun there, didn't they? Yeah. They defined everything about that. pH, amount of H+, right? Amount of positively charged hydrogens. We're back to ignoring hydrogen again. Okay, now the alpha carbon, you may have noticed there are 4 different things attached to that alpha carbon for most of these, right? There's an R group, there's the amino, and there's the acid, the carboxylic acid, and then there's a hydrogen, right? And so that's 4 different things attached. You may remember from previous chapters that 4 different things off of a carbon, these are called chiral centers, aren't they? And so there is, in fact, L amino acids and D amino acids. This is an L amino acid right here. And so you see it has these 2 on this side, and then the H and R. If we take the mirror image of that, that would be the D amino acids. Okay? Notice that this is a Fisher projection version. Do you guys remember Fisher projection? These are going back up here, right? These are coming out towards you, going across. Okay. Notice the naming is again the same as we talked about for the carbohydrates, right? And so it's on the left side, this last group right here. That's the L. On the right side, that's the D. Okay. Okay. Now, the L amino acids are the building blocks of proteins. The D amino acids do occur in nature, but are rarely incorporated into proteins, and they're actually quite rare in comparison. Okay. So the side chain or R group, this is going to give each amino acid its unique identity and characteristics. There are 20 amino acids that are going to be found in most proteins. There's going to be 9 families of organic compounds that are present. There's the alkanes, which are just hydrocarbons, right? Carbon chains with hydrogens. There's the aromatics, that's the aromatic rings that we talked about before. There's the sulfides, that's the SHs, right? And sulfur connected to things. Okay. There's the alcohols, those are the hydroxys, the OHs, right? There's the phenols. Remember phenol being the OH off of a benzene ring. There's the thiols. Thiols are just sulfurs, but alcohol-like. So SHs specifically, not S connected to something else. There's the amides. Those are the carbonyl, carbon-oxygen double bond, with a nitrogen connected to that same carbon. Okay? So like amine, but with a carbonyl. There's their carboxylates, like carboxylic acid. And there's the protonated amines. These are just going to be amines, but because it's physiological conditions, they're always going to end up protonated. Okay? So some of the properties of the functional groups are going to allow them to be classified as nonpolar and polar. The polar amino acids will be further classified as neutral, acidic, or basic. I know. Now you know why we had to teach you all the other material. It was all for this moment right here. Okay? The rest of the entire class was for this and the chapters that are going to come. Okay? And the little tiny things we taught you about physiology and along the way. Okay, so I'm going to give you this next part is going to be your amino acids. They're going to be broken down into classifications. Those are going to require some flip card work and going to require you to not quite memorize them perfectly, but you do need to have a decent idea. Okay? Now, keep in mind that there will be some questions that I'm going to ask you to, like, what are the amino acids here? And you have to identify them based on their side chains. You have to also identify what an amino acid is based on what, its structure is. Okay? So this part here, this is just amino acid, right? Period. Doesn't matter which one. See this purple part? That's the part that's going to be the specific amino acid. Okay? So glycine has a hydrogen on that R group. That's what the R group is. By the way, it also does not have a DL enantiomer. It's the only one that's not a chiral carbon right there because it has two of the same group. So that's glycine. The 3-letter code would be gly. The 1 letter would be G. And so they actually have 1 and 3-letter codes for each of these. If you know the full name, because I do multiple choice for everything, you could get away with just knowing the full name. Okay. For anybody that's going into medical, though, it's pretty common that people just expect you to know the 3 and 1 letter codes for these amino acids, and they will just full out give me gly, give me Ella. And they won't even say the rest of the name if they want. Like, they will shortcut it because they're just used to that. Especially doctors, because the MCAT loves to ask you questions like that. And they have to get it so well they can just breathe it out. They could do it in the middle of the night while they're sleeping. So this right here would be hydrocarbon. This is alanine. So he has the CH3 here. This would be valine. This one's kind of easier to remember if you see it because it looks like a V. Okay. This one right here is going to be another hydrocarbon. This is leucine. Now, isoleucine is going to be a rearrangement to that. See, it has that one down here instead. And so that's isoleucine. Okay. This is an aromatic one, and this is actually phenylalanine. Now, if you remember what phenyl means, you'll remember this one really easy. Because phenyl, meaning a benzene ring, right? As a substituent. Okay? That's what it is. Notice they've mostly used the organic naming. Mostly. Okay? This right here is a sulfide, so it's methionine. Methyl right there, right? Thio is in thiol, and so it's a methyl off of a sulfur. Okay? This is proline, and it's actually unique because it wraps back and bonds to the nitrogen. See that? Yeah, cool, huh? Okay, you all have a good day. I'll continue this next time. Next time we lecture, not next lecture, because next lecture is the exam. Please come prepared. You all have a wonderful day.